Welcome back, everyone. This is Ms. Bushra with you, a biology teacher from Zhyar English High School and Zhyar English Institute. This is our part 2 from the series of exam prep questions. We're going to start from chapter 3. Let's move on. We have question 21 here. Okay, it's talking about the Koch. Okay, Koch, you know that it's a scientist that developed a certain method called Koch's postulate in order to find out about microorganisms. So what did Koch do? How did Koch ensure that we had an isolated single species? We're not talking about the steps here. Isolated single species, when we use this word in a biological terminology, we're talking about separating a bacteria from all the other type of microorganism and make it a pure sample. How we do that? Let's see. How did he do it? It says by taking sample from an infected cow. Hear me out. If you take sample from anything, that sample contains a lot of bacteria, not only one. So it's not helping. Taking sample directly, it contains cells, it contains debris, it contains a lot of bacteria, maybe other type of microorganism too. So that is not isolating. Isolation is actually this one. By growing colonies of bacteria in the lab. We have something called Petri dish. Okay, Petri dish is filled with agar. So when we are going to, put our sample here in a certain method, striking, you end up with single colonies. Let's see. You end up with single colonies. These colonies are, developed from one bacterial cell that is going to be a pure one species of the bacteria. So we're holding on to this as an answer, but let's see why the others are wrong. By injecting them into healthy cows and extracting it. That doesn't help. You have a sample or you have something injecting it. Even when you extract it, again, it's a sample. It's going to be mixed. Another one, isolating the cow. Isolating the cow has nothing to do with the bacteria because the cow itself has so many types of bacteria in it. So that doesn't change anything. Our answer is B. In step 2 of Koch's postulate, we isolated in the lab by certain method growing colonies. Very nice. Let's move on. Histamine, complementary system, interferon. Again, this type of question. It gave you three things about immune system here. And apparently it requires you to find something that match them. This first one should be correct for histamine, the second one for complementary system, while the last one for the interferon. So we're going to look for that. We could start like this, from this line, and see which one of these is correct regarding histamine. Is histamine part of inflammation? Absolutely right. So this one is right. Is it part of allergic reaction? Yes. Histamine is part of the allergic reaction, we didn't know. So it is used in these two, type of reaction. Is it causing fever? No, it's interleukin, or sometimes the pathogen caused that to happen, or the macrophage. Is it part of inflammation? We already said it, so this is right. So in histamine, we excluded C for an answer. We don't want this anymore. Since one of them is wrong, the rest of it is going to go with it. That's elimination. Now we raise our chance to get a right answer. As for complementary system, complementary systems are a bunch of 20 types of protein. This is right. And they are ring-shaped. They puncture the infected cell. So this is right too, and this is right too. Puncturing infected cell, not puncturing pathogen or killing pathogen like this one. The second one was also wrong, by the way, here. All right, so all of the things here are correct. It doesn't give us any answer or any elimination. We have to find out our answer based on these. Interferon. Interferon is released by virus or creating resistance for virus or causing fever. Now interferon is like this. Let's assume that we have a bunch of cells, they are neighbors. One of them, this one, gets an infection from a virus. A virus went inside and it's dying. Before it dies, these are not selfish beings. What do? They create these interferons and release it. It reaches the neighbors and tells them, oh, I am in danger, you're going to be in danger soon. So what you're going to do, you need to try to build yourself to resist the entry of that virus. This way, they are going to create a shield for themselves and the virus cannot affect them. So interferon is not released by virus, but from the body cells of our own body to help other cells resist the viral infection. And it doesn't cause any fever. So our answer is B. Elimination, checking your knowledge, try to bring back the summary of what you've learned, even if it's through a drawing, it's going to help you. Now, moving on to question 23 and 24, it says a white blood cell that engulfs a pathogen. This word. You saw Pac-Man, right? The Pac-Man chasing the ghost. The Pac-Man, when it moves and opens the mouth, it tries to like chase it and then engulf it. Engulfing means like taking it in all at once. So white blood cells, some kind of it, when they encounter a bacteria, let's say this is the bacteria. I'm not naming the white blood cells yet, but we need to find out. It will engulf it. How? Oh, there is like a bacteria here. So these sides of cytoplasm will create something like an arm like that and encircle around the bacteria. And then they join together and seal it, trapping the bacteria here, then sending enzymes to the inside in order to kill it. This is called engulfment. Which one of these does that? Clearly, lymphocyte doesn't do that. Why? Because we have cytotoxic T cells, okay, that create what? That is actually puncturing cells, not killing bacteria or engulfing it. And then we have B cells that creates what? Antibody. It doesn't engulf. So lymphocyte is out. Macrophage is actually doing the engulfment, is the biggest one, and it engulfs it and also presenting the antigen of that bacteria or virus on the surface. That's how the cell-mediated on the humoral immunity start. So this is right, but we're going to keep it. Natural killer cell. Natural killer cells are not like what you think. Natural killer cells are killing infected cells on our body, and a cell that has virus already infected in them, it's going to kill that one for the sake of the rest normal ones, and kills cancer cell. By what? By puncturing them. Yani putting a hole in them, and when the water goes, they die. So this is not an answer. B and C are not option. Our answer is macrophage. Another one that is engulfing is neutrophil, by the way, okay? They're actually under one label called phagocyte because this process is called phagocytosis. Okay, as for question 24, receptor proteins are present in all of the following except. Receptor protein, if you remember it, they are proteins that can attach to the antigen and recognize them. So which one of the following doesn't have it? It says except. It didn't say no, but it said except. See, this here you need language, okay? We need English a lot for biology. Lymphocytes. Absolutely, lymphocytes are part of the specific immunity. They have these receptors on top of them that awaits any interaction with any antigen. Once there is an antigen attaching to them, it will immediately recognize it. So lymphocytes are not part of the answer because they do have it. RBC, is it part of the immunity? No. RBCs, unlike the rest of the cells that participate in the immune system, they don't need to recognize the antigens. So instead, they have antigens themselves, but no antibody. no receptors for it, okay? So no receptors in the RBCs. So RBC is the right answer, but let's check the other. Antibody is a receptor itself. It has a Y shape like this, and both ends here are receiving and binding to the antigens. As for macrophage, most of students think, oh, a macrophage is a non-specific immunity. They cannot recognize the type of the antigen. Yes, they cannot recognize the type, but they do know this is not a body cell and this is a foreign body by receptors. So these are receptors too. The answer is RBCs. So red blood cells don't have any receptor protein. We have this question, please screenshot it. It's very easy, it's just recalling from memory. Okay? Check this out. We have numbers here. If there was more numbers, it was going to look a bit more intimidating than normal. But what we're gonna do, we're choosing the easiest part that we know. I for sure know which one is viral antigen. That's the easiest for me. So it's 1. And since there's only one here, that means A is the answer. But let me check the rest of it before we decide. Cytotoxic T cell. I know this is an infected cell. This is a macrophage. This is a virus. This is T cell. And this is cytotoxic T cell. So number 2. That means number 4 is out because it's not cytotoxic T cell. Number 4 is actually T helper cells. So 2 and then 3. What's supposed to be 3 or 5? Interleukin 1. Interleukin 1 is released by macrophage. This is interleukin 1 released by macrophage. While interleukin 2 is released by T helper cell. This is interleukin 1 and this is interleukin 2. The question asked is about interleukin 1. So we have to choose which one? 3. 3, this is the right answer. Even if there's 10 labels here, don't panic. You just go one by one or choose the one that you find it the easiest to look for. That doesn't work entirely with the steps. If they're steps following each other, these are separated from each other and comes from a label, so it's easier. All right, so in question 26, like I said, don't get panicked when you see this graph, but actually, we're just looking for keywords here. It's talking about the shorter duration of second immunity and what's the reason behind this response. You see, this is primary immune response and secondary. There's less amount of antibody created here, more here. And the time until you reach to create antibody is longer than this time in the secondary one. Why all this? What do we have in the secondary response that we didn't have in primary? Just as a recap, secondary response is when you already had the disease once before or twice before. Your body built an immunity. So what causes this immunity to stay, okay, to protect you for a very long time? Is it because mutation made by the body? No, mutation doesn't help you like that. It's a natural process, already happens in everybody. It's not for one person. If it was a mutation, some people would have it, most of us wouldn't, because it's something randomly happening. Is memory cell development? Actually, this has a huge part in it. Why? When memory cells are created during the humoral responses from the B cells, these have all the information about the previous disease. So they don't need to, spend time recognizing that. Once they encounter it, they say, oh, that disease, we already know what type of antibody to create for it. And they create that antibody and release it immediately. They already have the structured antibody and they know how to do it. In the previous one, when it's your first time, you don't have the specific shape of the antigen or the disease-causing pathogen. So they have to analyze it and try to fit and make a shape that is going to fit with it. Make a shape for the antibody. So macrophage recognize pathogen. That has nothing to do with immune, secondary and primary, because these are part of the specific immunity. This has nothing to do with specific immunity. Fewer pathogens entering the body? No. Even like if there's few, but if the memory cell encounters them, there will be a reaction, okay? Has nothing to do with that. So the only reason why secondary is shorter and stronger, it's because of the memory cell development during the first time. Memory cells are developed from the first infection, but they don't act there. But in the second infection and onward, they are recognizing the pathogens, and they are the one that produces the rest of the cells to fight it off. Very nice. We're moving forward. Question 27. It says, which of the following is not correct? Okay, we're looking for something that is wrong. According to what we see here, it's all about chapter 3. Asthma here, for example, is a reaction against harmless antigens. Yes, we have 3 immune problems that are, I call it personally, triple A's. Asthma, allergy, and autoimmune. Each one of them is not due to an infection. It's actually something harmless that causes that. Asthma is one of them. So this is something correct. That means it's not our answer. Vaccines contain antibody to protect the body. This misconception is everywhere around. Vaccines don't have antibody for protection. Vaccine protecting you by putting antigen, or you can say the pathogen or part of it, into your body. That pathogen or antigen is weak, or there's like one section of it, one part of it, or it's killed, heat killed. Why do we do that? If we put an antigen that we're sure that it's not going to cause, damage into your body, but we can trigger your immune cells to respond to it, you're going to pass primary. So when the actual disease come, when the actual virus or bacteria come, your body already built memory cells and it can fight it very quickly, very shortly. They develop the antibody. So it's a trick. We're going to inject you in order to develop immunity naturally in the body. So this is actually wrong. This is my answer, but let's see if the others are correct, just in case. Histamine is released during allergic reaction. Yes, in 2 cases histamine are released, allergic reaction and inflammation. Multiple sclerosis may cause blindness. Yes, multiple sclerosis is an autoimmune disease that is attacking the myelin sheath of the optic nerve cells or optic nerve neurons. So this is actually right. This means not our answer, and we have B as the right answer. Moving on to question 28, it says, why can't HIV infect helper T cells at the beginning of the infection? We reach section 3 here. Let's see why. See, helper T cells, okay, are not the target for a reason. It's not because the HIV doesn't want it. Actually, this is the bigger, target for the HIV, but it cannot. Why? Because for HIV to enter any cell, entering is a must, okay, it needs a co-receptor CCR5. We have 2 receptors, okay? One of them is CD4. Both macrophage and T helper cell have it, but CCR5 is present on the macrophage where HIV can attach to it and enter it, but the T cells don't have it, so it cannot go inside the T cell. What's happening is that when that HIV is developing within that macrophage, sometimes there is a mutation. It takes a very long time, but there will be mutation, and that mutated virus is going to make more offspring later on, making sure that these offspring have all ability to enter T helper cell. This T helper cell, on the surface of it, it has CD4, that's T helper, by the way, T helper or T cell, and co-receptor other than CCR5. Okay, other than CCR5. So let's see, what does it say? Why can't it infect it? Because it doesn't have CCR5 at the beginning. Okay, T cells lack CD4. No, they do have it, so this is not the answer. T cells have strong defense mechanism. No, it has nothing to do with that. T cells lack CCR5 co-receptor. That's the correct one. Okay, they do have CD4, but they don't have CCR5. One more time. CD4 is for binding and recognizing the cell, but CCR5, for the virus to go inside the cell, it needs that co-receptor. Got it? Very good. Question 29. See, when is a person considered to have AIDS? Most students have this misconception thinking that once an HIV or any virus enters the body, done, you're infected. No. If there's no symptoms, if there's no effect, if your defense got it eliminated, it's not causing any disease for you. So it's not considered as a problem. It's a bit more special because sometimes HIV stays in the body for 10 years before it develops any symptoms and creates AIDS. AIDS stands for acquired immunodeficiency syndrome. Means that person's immunity is going to be so low, no longer can defend itself. So upon entry of the HIV is not a correct answer. You don't have it immediately. When the T cell count drops to 200 cells per milliliter, this is from the book, this is the major sign of having the AIDS, the onset of AIDS, and it's the third phase. But sometimes, since we have very low immunity, another thing, even before we check this, another thing could tell us, which is opportunistic infection. Infections that you don't get it on normal body immunity. Your body immunity, if it's strong, you don't get it. Okay? So these type of infection only appears when you have a damaged immunity, and this is where you can find AIDS as well. This could be the name of the phase as well. These two are correct. B and C are the right answer. We're on to the last question of this chapter. We're talking about HIV is transmitted by transfer of body fluids containing what? This is a question from Wizari last year. The majority of the students have a problem with it and they mistake it. Probably already solved it and it's easier now, but we're just gonna remind you one more time so that we don't get to make that mistake again. See, if you have the virus and the virus directly enters the body, it causes infection. For example, HIV enters your body, it causes infection. So it could be transmitted like that, fluids have that. If you have a body cell, this is my body cell for example, okay, and it has virus within the cell, that virus is also going to come out when it enters the body of another person and cause infection. So in both cases, as for this is anti-HIV antibody, that has nothing to do with the infection. It's just an antibody against the anti-HIV. This is used for test. So these two are correct. In order for us to choose it, D is the right answer. That's how we got it. We're done with questions from chapter 3. We're entering chapter 4. In chapter 4, most students think this is the hardest part. No, it's most detailed and you need to focus on your answers. Let's try to grasp as much information as possible from these questions that I put it here. It says, which of the following structures is not found in neurons of the central nervous system? Central nervous system means brain plus spinal cord. Now, we have neurons like this, typical neurons. This is axon. These are dendrites. There are many and they're shorter. This is the nucleus in soma. And these are myelin sheath. What is this? Myelin sheath. Okay? Myelin sheath is created usually by other cells that surrounds and insulates the neuron. What is interesting is that every part is present in neurons of the brain and spinal cord other than this. Okay. But does that mean that neurons of spinal cord and brain don't have myelin sheaths because the Schwann cell is the one creating the myelin sheath? No. Myelin sheath is present there, but it's created by other cells that are not Schwann cells. Got it? There are cells, we didn't name them, but they are the one creating it in that area. So the only part that is not present in the CNS is actually Schwann cells. You could never find it. The only places you can find it is outside of that in the peripheral nervous system. That's the only thing we grasp it here. We have question 32 here. It says, what accurately describes the ion movement during the phase ion movement, don't forget, of 3, this one. We're looking for that one. I know it looks intimidating, but let's start it simple. Let's try to label. Number 1 is depolarization. Okay, number 2 is repolarization. Number 3 is refractory period. And this is, when we like going back to its original, the membrane potential. So here, if we look at 3 in the refractory period, we know that since a lot of sodium, if this is the cell, during the depolarization entered the cell, sodium must not be inside the cell normally. And during the repolarization, potassium, which is supposed to be inside to bring back the membrane potential, the resting membrane potential actually, okay, it is outside. So it's switched. You have more Na in and less K inside. We need to switch back this to normal. The polarization of the electricals are correct, okay? The outside is positive, the inside is negative in the refractory period, but the concentration of the molecules are not the same. So what we're gonna do, we're gonna switch back these two slowly by a method of using sodium-potassium pump. Which of these says sodium-potassium pump? These two. These are not correct because refractory period doesn't use sodium ion channels nor potassium ion channels, but it's using sodium-potassium pump. That pump brings 2 potassium inside and throws 3 sodiums outside. This is how it regains back its polarity and its concentration of the ions. I'm going to write this, but what does that have to do with this question? Nokia. It was like an old phone, if you remember, but it's going to help us to answer. Instead of me trying to figure out, oh, what was it going out? What was it coming inside? This is a very short way of answering. Pump, the sodium-potassium pump works this way. Na out. Think of it like that. K is for potassium. Okay. Nokia, potassium. In. And it's using ATP. Once you memorize this, it's a piece of cake. I don't know why this is bothering me. That's how you know. So Na out, K in. Which one says that? K moves out? No, it says moves in. So this is the right one. And Na moves out? Yes. Using the sodium-potassium pump. That's how we got our answer. Easy peasy, lemon squeezy. All right. It says stimulation of the nervous system is through what? Electrical impulse within cell, or chemical signals between cells, or both of them, or none of them. See, if you have 2 neurons that they're communicating, let's draw neurons. This is the first one, and this is second one. Let's make it black, okay? They are communicating with axons and dendrites. You see, electrical impulses are only in axon. Just like a wire. If you have a gap here, it doesn't conduct the electrical impulse, right? So here it's electrical. Once there is the gap of synapses here, we change it to chemical signal. The chemical signal is neurotransmitter. That chemical signal is going to be read here again, and they're like processing it. They make it into another electrical impulse, okay? So it's electrical, chemical, like that. Within the cell and the axon is electrical. Between two cells is chemical signaling. That's how they do. And that's how we got our answer. It is C. Okay. This is another questions of Wizari that a lot of students have problem with it, and they even chose two answers just because of a keyword that was used. So in that one, students were asked to find areas where mechanoreceptors, you know, mechanoreceptor is a type of receptor that responds to physical movement or physical touch or whatever. It doesn't respond to chemical or electrical or pain. It is physical. So here, the thing that gave everyone away was using hemisphere instead of brain, because the label 3 is cerebellum, while these are all part of the cerebrum. So we're gonna consider that this is talking about brain and try to answer it based on what we're saying. Area number 1 is parietal lobe. Parietal lobe is known for sensing. Every sensory information goes there, and sensory like touch. Touch is something physical. There's no chemicals here. There's no light. There's no anything else. Touching is going here. So it's mechanoreceptors that read it and send it here. As for second, it is the temporal. Temporal brings back signals from ear. And we know that the ear has the hair cells that responds to the movement. So 1 and 2 are both correct. What about 3? 3 is cerebellum, and cerebellum is the side where all the information from the muscle positioning, tension, and everything comes back, and it's all due to mechanoreceptors that these signals reach us. So the answer is 1, 2, 3. All of them have mechanoreceptors reaching out to them. This is how we got our answer, 1, 2, and 3. As for the last 2 questions, I'm gonna leave this in my channel on TikTok. I'm gonna release it soon. A reminder of how to contact me or see my videos on my TikTok. This is it for today. Tomorrow I'm gonna see explaining more questions regarding chapter 4, and we're gonna continue with the rest of them. Thank you.